I think the one area that I'd be interested in Hari's input as well, and this has always had, it was challenges for me in the past, in the days of biologics, is exploring incoming raw material variability. So if you think about polymerases and complex enzymes, et cetera, I think that's one area that I would say there's a bit more work that needs to be done to understand, those potential sources of variability and how that impacts your deterministic. I love that terminology, the deterministic IVT reaction that you can measure.
That's a great point. Yeah, go ahead, Hari, and share your thoughts.
I think on that particular point of raw material variability, absolutely important. We use enzymes in these processes. While it's not a cell-based process, you still use a biological entity such as an enzyme, which can have some variability within itself. So, properly characterizing that is extremely important. The good news is that we've spent 40 years now characterizing proteins, so we know how to characterize proteins really well. So we can apply all of that knowledge, frankly, on these enzymes, which typically have not been characterized to that level. Because they are raw material. They're not. Typically, people don't do the same level of characterization as you would for a therapeutic, for example. But we have all the tools. The rest of the characterization is even simpler because they're all small molecules. DNA plasmid, of course, is different. That can also be characterized quite well because DNA has been used as a vaccine candidate and as a therapeutic candidate. So there's a number of tools and techniques that are very well developed for characterizing that. And I think, so ultimately, characterization of these input materials is important in controlling the variability of IVT. On the question of the lowest hanging fruit that you had asked, I think the obvious answer there is, figure out which ones are the most expensive raw materials and figure out, optimization around that, right? And what they are depends on the kind of process you're pursuing. But optimizing around the use of your most expensive raw materials is the lowest hanging fruit.
Absolutely. Keeping cost of goods top of mind from the very beginning, right? Thinking about that. And I like this notion too of, one of the biggest challenges I've heard from folks in the space is navigating the supply chain and some of the quality differences, right, that can occur, or the lack of even just harmonized documentation around the quality validation of those materials. And so, John, I'm curious, did Hari help with your question, right, in terms of, do you feel like you also need to be, do firms need to be implementing or developing more internal standards, right, for quality at this point in the supply chain and in the industry? Or is this something that, you've had to implement in-house activities, right, to validate these materials more carefully, or invest more deeply in the analytics of these? Is that something in your experience that you think firms need to do more of, or that you've had to do in your experience?
I would say yes. I think these are always the wounds of the history that we've lived here, right? Hopefully we're learning organisms and we get smarter as we go along. And I think as Hari pointed out, right, when you're looking at more biological processes versus, I'll say, more synthetic processes, right? And when you look at the evolution of the analytical techniques that are out there, right, it's just we have so much more capabilities today than we have had ever. Now, saying that as well, it's important that the full supply chain, and when you look at the suppliers, there's good reputable suppliers out there. You have a good supplier management program that's in place, right? So we don't take on all the work, right? Companies can't afford to take on all the work, and there's a reliance on the supply chain, good suppliers looking for secondary sources and doing very robust technical analyses and assessments around their processes, and ensuring that there's good quality systems that are in place as well, right? And it's going to be an evolution. We certainly use reagent grade materials early on, and we do the appropriate assessment of that material, and there will always be this transition to GMP grade over the course of time.
Yeah, phase appropriate approach. Yes. I'm curious, are you finding any pressure to move earlier to GMP? Is that something that you're seeing from a regulatory perspective? Is that, or has it stayed phase appropriate? You feel the pressure?
From my perspective and recent interactions from the agency, I think there's always a reasonable expectation on the evolution. Again, when we say phase appropriate and we look at the denotation of GMP versus reagent grade, we are doing a level of characterization and risk assessment around our reagent grade materials, right? And it's appropriate, and frankly, the data needs to be package needs to be strong, and you need to stand behind it.
Yeah, absolutely.
And so if you have that, and you take a good scientific approach to this, then it's a very logical approach. And I really have not seen, in my experiences at least, any differences in approach than I had taken previously with biologics or vaccines.